Liz, welcome on in to the SciAnts colony. Thank you so
Thank you.
much for joining us today.
So I am Dr. Elizabeth Marnik. Most people call me Liz. I have a Bachelor's of Science in Biochemistry, and then I did a PhD. And my PhD is technically in genetics, but my research used genetics to understand the immune system. So my project was all looking at how the immune system functions normally, and then how it gets messed up in individuals who have autoimmune diseases. And then I left that work for a little while, and I was an assistant professor where I taught immunology and microbiology. And now I'm doing K through 12 science outreach work and public outreach work at a small research institution on the coast of Maine.
Awesome. Thank you so much for joining us today, Liz. So we've been kind of going over the incredible guest journeys in the sciences, like what their journey has been like and what it means to them to be involved in the sciences. So let's just take it back to the beginning. What attracted you towards the sciences? What was it that made you be like, this is what I'm interested in and this is what I want to do?
That's a good question. So my journey to science is kind of strange because I was raised in a very conservative family. So I was raised fundamentalist Christian, and I was taught that my job was to get married and have babies. Those were the two things I was supposed to do with my life. So I wasn't vaccinated growing up. We didn't do any sort of like medical interventions. I don't remember going to a doctor until I was like a teenager. So it was interesting because I was homeschooled because my parents didn't want the public school system to corrupt me. So I finally had my first science class in 11th grade. And this was after I begged my parents to let me go back to public school because I was teaching myself by this point. In 10th grade, I literally assigned myself essays. I graded my own essays and I assigned myself my own work because nobody else cared. So I finally go back to 11th grade, and that's where I had my first science class. And it was then that I fell in love with science. And then I had some really good teachers who taught me, I didn't even know how to apply to college. I didn't even really understand what I could go to college to do. And it was because of those teachers in 11th and 12th grade that helped me do that. And then I ended up going and getting a 4-year degree in biochemistry. And then once I was in college, that's kind of where I fell in love with research because I worked in a lab as an undergrad. I got vaccinated for the first time in my 20s. And yeah, from there, I would say that it was discovering what science was and the cool things that I could do with it that really made me decide in college that I wanted to go get a PhD.
How difficult was that transition of, like you said, growing up in the household where it was like you're being protected from that kind of system to now being an educator of the sciences? It seems like it's two very different sides, right? Like, how has that transition, do you think, affected you? And what would you say to folks who are maybe curious about that possibility, but not necessarily, they're feeling maybe nervous about it? What is your advice to someone who wants to go through maybe that, just even dipping their toe into the sciences?
Yeah, I think one thing that was really important to me now is that people realize that science is for everybody, right? We think of science, I think, sometimes as only being in a lab, but the reality is that science impacts people at all stages of their life. If you took Advil today, for example, that was thanks to science. So I think from my own experience, I really learned that exposure to science information can really change someone's life trajectory. I probably would have been married and had a baby in my 20s otherwise. I do have 2 kids now. I am married now. I have a career. I have all those things. But my life would have been very different if a teacher hadn't shown me the possibility that was science for my life. So I think one thing I like to remind people of is that science is for everybody. You can just go explore nature. You can go on a hike and see what you see as you're hiking around, right? Science is for everybody. And at some level, science impacts all of us. So even if you're intimidated by the idea, it's never too late. When I was teaching at the university, I had students in my class who were 55 who were going back to school. Or I had one time I even had somebody who was 70 because they were like, hey, I didn't think about doing this sooner. I raised my kids, did all that sort of thing, and now I want to go learn science. So it's never too late to do that.
I think that's really, really cool, especially that approach that science is for everyone and framing it in the way of going outside is science too, like participating in just like day-to-day activities. It doesn't have to be something like extreme, like the biochemistry and mixing chemicals. What got you interested in that biochem side of things? So you went off to college and you found the biochem life and lab life interesting. How did you go into your first lab, and why did you pick the topic that you were interested in?
Yeah, so I actually entered college as a chemistry major because it was that high school chemistry class that I took in 11th grade that like blew my mind about how cool science could be. So I entered college as a chemistry major, and then as I started taking classes, I realized that I really liked learning how living things worked. So that's why I ended up transitioning to biochemistry. So I ended up switching my major at like the end of my freshman year of college, because I really wanted to study how living things worked. And then from there, I took a genetics class and I fell in love with genetics. And the professor who taught genetics ran a lab, and he saw how excited I was. So one day he pulled me aside after doing an experiment in the lab, and he was like, "Why don't you come work with me?" And the school that I was at actually required everybody to do 1 semester of research to graduate. So I needed to do it anyway. And he offered a spot in his lab. And then from there, I just fell in love with he worked with mice. So I did mouse genetic work for my undergrad. And then I used mice again in my PhD. But it was getting into the lab and realizing that really kind of blew my mind.
And it seems like both times you had like a really positive interaction with the professor that led you towards that. So, we hear a lot here and other places that I really used to like science, but I had this negative interaction with a teacher and they made me not like it anymore. With your experience now, like what would you say are some of the winning factors that kind of keep people in the sciences? What are some things that could, instead of driving folks away, it would be keeping them in there?
Yeah, I definitely think for me, it was the support that I found from professors, right? Because I grew up in a family where, in reality, I don't think they understood why I decided to go to college in the first place, right? Because I should have been getting married. But it was a different experience for me, because when I got there, I started meeting new people who were outside of that bubble, right? I grew up in a very isolated bubble. I was telling somebody the other day that I don't get a lot of cultural references because I wasn't allowed to watch TV or to listen to secular music. So a lot of those things sometimes like go over my head because I wasn't a part of them. But when I finally got into college, it was really building relationships with my peers who were in those classes with me, and then my faculty members who really made it easier for me. And there was a point where I was a senior in college and I was going to drop out because I got to the point where like I was taking care of my mom, I was taking care of my brother, there was a lot of family stuff going on, and I didn't see a way forward where I could keep doing what I was doing. And my professors noticed something was going on, and they essentially staged an intervention. So a group of them were like, "What's going on, Liz? What can we do to help you?" Like blah, 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 blah. To make a long story short, they helped me move out of my house. They helped me get a car so I could take it with me to graduate school. And they really like gave me the resources to keep going. And I think if I hadn't had them, I would have quit, because I was at the point where I couldn't see a way forward, and I couldn't keep doing what I was doing because I was working full time. I was taking care of my family, and I was in college. So I think when we think about it, I think we really undervalue those professors and those teachers, right? It was really, if I have to credit anyone, it would be my 11th and 12th grade science teachers, and it would be the professors that I met. So I think valuing that kind of work is more important now than ever, and making sure people who are in those positions have the training, because not everyone is good at mentoring students or supporting students. I also had some bad professors, but luckily I had more who were good, right? So I think really understanding that you can be smart, but if your life circumstances aren't allowing you to keep going, then you're going to lose those people, right? So we have to set it up so that people have the support they need to keep going.
I think that's a really, really cool response that I think can resonate with a lot of people, because it's not always just this easy road of like staying in a particular field, and the accessibility to the field then, right, is really limited for a lot of people. What made you decide then, when you were continuing on, like at the end of your undergrad, you were in this lab working with mice, to go to graduate school and to keep with mice? How did you decide that? And tell us a little bit about the the research that you did as a graduate student?
Yeah, so when I was an undergrad, I worked, as I said, in a mouse lab. And if you buy mice in research, you probably buy your mice from the Jackson Laboratory or maybe Charles River. But Jax is what we call it. They make mice and they sell them to researchers across the world. And the lab that I was in as an undergrad, he had done his postdoc at the Jackson Laboratory, and that's where we bought all our mice from. So as I was exploring graduate schools, I knew that I wanted to do something with mice because it was what I had done and I liked that work. And I think that felt safe to me because it was like something I knew. So I started looking at PhD programs, and the Jackson Laboratory had just launched a new collaboration with Tufts University. So I was like, oh, I'll apply to this. I was like, we buy our mice from Jax. It would be kind of cool to do my research there. So I applied to about 10 PhD programs. I got into 4 out of the 10 that I applied to. And I ultimately decided to join this new program. I was the first student in this program through Tufts and the Jackson Laboratory. And I did it because I figured if I want to do mouse genetic work, there's nowhere else better to do it because they are the place to make the mice. I had a great experience interviewing there. I really liked the faculty that I had met. So I decided to accept that offer. And I started that program. And in grad school, you do rotations. So you get to test out different labs. So I tested out a bunch of different labs. And I ended up joining the lab of Derry Rupinian. He is now retired. I was his last official grad student. And he actually had worked with my undergrad mentor when he was at Jax. So it was kind of like this full circle moment. He had helped train the guy who trained me as an undergrad. And I really liked him as a mentor because he was very supportive. He was actually my first rotation as a grad student. So it was a really rough transition it was my first, I moved to Maine from Connecticut. I'm from Connecticut. So it was my first time ever moving away from the place that I grew up. So it was a big transition for me, but his lab was really nice. His lab was very supportive of me. The research question was really cool because we were using genetically altered mice to study the immune system. And by that point, I really liked figuring out the immune system. So I liked the question. But I left his lab and then I went to do other rotations because I had to do a couple in Boston too. So I kind of split my time between Maine and Boston. And I had some really bad rotations where the person running the lab was not supportive and was not willing to answer questions. And I almost quit grad school. And Derry sent me an email and he was like, I just wanted to check in and see how you were doing and let you know that you did a really great job in my lab while you were rotating. And there's a spot here for you if you want it when your rotations are done. And I was like, okay. I was like, so maybe I don't suck at this. Because at that moment in my rotation, I was like, oh my God, I'm terrible at this. This is not going to work. So yeah, I ended up joining his lab once my rotations were done. And it took me I did my PhD in 5 years. And my work was focused on, so in your immune system, there are a lot of different cells. I like to think about, there is the innate immune system, which is, I like to equate it with your first responders. So your EMTs and your police officers. So if you're in a bike accident one day, those are the people who respond first. And it's your innate immune system that responds first if you're exposed to SARS-CoV-2, which is a virus that causes COVID, or measles, or whatever virus it is. Your innate immune cells respond first. And then you have your specialized cells. Those are your doctors and your orthopedic surgeons and all of those specialists who can help take care of you if you broke your leg. And your adaptive immune system has 2 different kinds of cells. It has your B cells and your T cells. And I studied a type of T cell called a CD4 T cell. And my work was all trying to figure out how does this type of cell function? And we can get even more specialized than that. There's a specific kind of CD4 T cell that I studied, and I looked to try to figure out how does this cell function normally, and then I had mouse models of autoimmune disease. So then I was like, well, how is this cell type altered in mice with rheumatoid arthritis and mice with lupus? And that was what I spent my 5 years studying, trying to figure out how does this cell work normally, and then what are the things that are different in the context of that disease.
How easy or difficult it is to work with mice. So we have a lot of questions from the community of the organism in which you work with. How is the day-to-day look for something like that?
It is hard because they're mice. It's funny because working in the lab with mice, it didn't bother me. But then if I came home and I found a mouse in my house, I would freak out. And my husband, like we were dating at the time, he was like, you work with mice all the time. He's like, what's the deal? And I'm like, a mouse in my house is very different than a mouse in the lab, first of all. But yeah, it's hard because they bite and they pee on you. So you're working, we wore protective equipment so that we wouldn't get them sick, but then also so that they wouldn't get things all over us. And whenever you're working with an animal that has a spine, you have to make sure you're following ethical procedures to make sure you're minimizing pain and suffering to the animal. And so there was very strict rules, which there should be for that kind of research. So I took blood samples a lot, for example. So I had to make sure that I was using like a numbing agent to take the blood samples so that I wasn't causing a lot of pain and suffering and those types of things. So it's definitely tricky to work with them because they are a living organism, right? And compared to some of the other ones you can work with, they're more technically challenging because they can bite. Whereas I now work with C. elegans, which are a little tiny roundworm. They don't have teeth. They don't bite or pee on me, which is lovely.
I've never heard it as the opening issue of they bite and pee, but it's true, right? Like that is something to have to account for those differences. Question from the community is, can you go into a little bit about the differences of T cells? So what are the different types, and then what was the type that you were working on, and why is that type interesting?
Yeah, that's a great question. So there are two kinds of T cells. There are a category that's called CD8 T cells, and we call those cytotoxic T cells. That's the other name you'll sometimes see them referred to. And then we have another kind of cell, the CD4 T cells, and we call those CD4 helper T cells. So for CD8 cells, they have two really important jobs. One is to kill any cell that's abnormal. So they help survey your body and kill a cell that maybe is turning into cancer, for example. But the other really important thing they do is when a cell has been infected with a virus, their job is to kill that cell that's been infected with the virus. And that's because viruses are different from other types of pathogens in the sense that they can't do anything on their own. So to copy themselves, to reproduce, they have to infect a host cell and hijack that host cell. So CD8s, they have the job of identifying those hijacked cells and then destroying them so the virus can be eliminated. That's why we call them the cytotoxic T cells, or sometimes they might be called the cytotoxic killer cells. The other category of T cell are the helper cells, the CD4 cells, and their job are to help other immune cells do their job. So that's why we call them helpers, because they are the supportive cells to make sure other parts of your immune system can function. So I studied a type of CD4 known as a T follicular helper cell. And we name, immunologists like to name things in ways that are very weird sometimes. But their job is to help B cells make antibodies. So you guys may have heard of antibodies. I don't think I have it in this room. I have a stuffed animal antibody that somebody gave me. But it's the little Y-shaped molecule that your B cells produce, and they circulate throughout your body and they bind to pathogens and help destroy them if you get exposed. So your B cells produce those antibodies, and the T cell that I studied, it has the job of supporting the B cell so that it can do that. And we have research that shows if the T cell isn't around, the B cells don't produce antibodies the same way. They don't do it as well. But in the context of autoimmunity, the problem is that you're producing antibodies that are attacking your own tissue. They're attacking itself. So one of the questions I was trying to answer is, why does that happen? And is it the B cell's fault? Is it the T follicular helper cell's fault? Or maybe it's a combination of both. And in reality, it's a combination of both of those things that are happening.
Is the follicular in the name referring to some kind of follicle cell in a particular location of the body?
Good question. Yeah. So that stands for, yes, it's a follicle. So in your immune system cells kind of hang out in different places. One is your lymph node, one is your spleen, then your blood, and then sometimes your bone marrow, depending on the kind of cell you're talking about. So follicle refers to an area in your spleen and your lymph node where your B cells are copying themselves and pumping out antibodies.
Okay, so it's like a home hub for those cell types.
Yeah, so like after, you might notice that like I heard you just got a couple vaccines, right? So you might have noticed, and sometimes this happens, you might get swollen lymph nodes. And the reason we get swollen lymph nodes after we get sick or after we're vaccinated is because those are the areas where your immune cells will copy themselves and start pumping out those antibodies so they get bigger to accommodate the fact that there's more cells that are copying themselves in that location.
And that's why there's that pressure too. Like if you start touching the lymph node, you start feeling that like they're a little bit on that bigger side right afterwards.
Exactly.
It's really cool that you can feel the physiological output that they're like priming themselves. Like that's just a wild thing to think about. Like it's really, really cool.
It is cool. And when I remember when I got my first COVID shot, I was working at a vaccine clinic during the rollout. And I remember like I got it, and then like 3 days later, I noticed my lymph nodes were a little big, and I was like, yes. I was like, my B cells and T cells are working.
So it's just as a total aside rabbit hole. Yeah. Is that an indicator that the vaccine's kicking in?
So this is the hard part, right? It's not, if you don't have that noticeable, if it's not noticeable to you, that doesn't mean it's not working. Some people just react stronger, and they might have we call them reactive lymph nodes than others. I am one of those people who every time I'm sick, like I can just have a normal cold and my lymph nodes will get big. So it doesn't necessarily, if you don't notice your lymph nodes swollen, don't panic. That doesn't mean your immune system's not working. It's just some people will have noticeable changes and others won't.
Back to a little bit of that genetic variation between individuals.
Exactly. There's that whole spectrum of people. This is why some people get sick very little. Some people still have never had COVID, right? So now researchers are trying to understand, well, what is it about them that maybe makes them resistant? There's this whole spectrum.
For me, it's staying inside and not going anywhere.
That helps too. Yeah.
So what was, in your studying like these cells, what was the coolest result that you ever did or ever got? And what was the coolest technique that you ever did to you? Like, what was it that you were like, man, this is so cool to be able to look at and see?
So I think one of the coolest results that we got was actually we were using a mouse model of rheumatoid arthritis. And we were actually able to show that if you change the microbiome, so microbiome, for people who maybe don't know, is if you think about it, we have our own cells that are making us up, but on our skin and in our guts, everywhere, we also have all of these microorganisms. We have bacteria, we have fungus, we have all of these other things living inside of us and on us all the time at this very moment. And the microbiome refers to that whole combination, the bacteria and the fungus, all of those things that are living on you and in you. And we're starting to learn more and more that those things actually do have potentially an impact on our health. And we had this mouse model where we gave it antibiotics to completely wipe out its gut microbiome. We gave it like crazy high doses of antibiotics. This is not something you would want to go do, please. And we noticed that their symptoms like got better. Their swelling of their joints got better. And I was actually able to see that the cell that I was measuring was decreased compared to what it was when they had the swollen joints and stuff. So it was really cool to see that there seemed to be this feedback between the microbiome and triggering or worsening these autoimmune symptoms. And we don't know what it is still, right? Researchers are still trying to figure out, is it the autoimmune disease that changes the microbiome and then it gets worse? Or is it the microbiome that is it the chicken or the egg argument is always happening? But it was really cool to see that there was this direct effect of the gut microbiome on the symptoms that this mouse was having. That was really cool.
That's an exciting result, and also speaks to like potentially other side effects that could happen too, right? Like you've cleared the gut microbiome. So like, yes, you've helped the arthritis component of it, but now what are the other issues that you've caused? And so it's always like, it makes it really difficult, right, to study these questions because it's just all one giant web, and you can't really study everything one at a time, especially in something as complicated as a fly, but let alone a mouse.
Yeah, right. Because like in humans, we know that after somebody has taken a lot of antibiotics, like for Lyme disease or for another infection, sometimes you can then get like an infection called C. diff. And it's where, because your microbiome in your gut has changed, a bad bacteria take over and then cause like severe symptoms. And now one of the treatments for that is actually fecal transplants. and they're now doing fecal transplants for those people to try to, so they can reintroduce the good bacteria to get rid of the bad bacteria, is essentially what's happening there. But yeah, those are the questions. So we can do it in a lab in a mouse, right? Because we can control it better. But it takes a long time for this work to eventually get to a human, because we don't live in a sterile environment. My mice were in a room, right, that was not perfectly sterile, but was not outside in nature. So it's easier to study those questions in that kind of setting than it is in a more complicated human environment.
Question from the chat is the gut biome causation or correlation in that rheumatoid arthritis model?
Yeah, so that's a great question. We had some evidence that it likely was causative, because we were able to actually introduce individual bacteria species back into the mouse and actually re-cause their symptoms.
That's really cool. So you could figure out which bacteria were the ones that were in combo.
Yeah, there was at least one. Then there could have been more, right? Like we reintroduced one kind of bacteria and saw that the symptoms appeared again, and the cells that I was measuring got worse again, like increased in numbers again. But it could also be other ones that would be playing a role too. That could have just been one of multiple that would have that effect.
Really cool. And so that was what the really exciting result that you got. What about in terms of a method that was just, for example, our community really likes anything imaging. Whenever we do some kind of microscopy in the paper that we review, everyone's like, whoa, it's real cool to see. What was that whoa, that's so cool? So
we were able to do some really cool bone marrow transplant experiments with our mice, because we were trying, so your bone marrow is where your immune system cells come from. So we were able to do some really cool experiments to look at the genetics of what was happening by having one mouse that had one combination of genetics and then irradiating that mouse to get rid of its bone marrow, and then giving it a bone marrow transplant with a different type of genetic, cells that come from a different background, that had a genetic mutation that we were interested in. And then we were able to study, well, what was that impact on the immune cells that mouse then started to make because of those differences. So it was really cool to be able to do that, because that's not something we would ever want to do in a human for lots of ethical reasons. But it allowed us to answer really cool questions about how different genes that are involved in the immune system can help different cells function.
That is really, really cool. And presumably you can test in a couple of different ways, right? You can test through visual markers, you can do immunohistochemistry and see how the cells are behaving. You could do I guess you do inductions of infection and or just checking the inflammation levels for the arthritis too. So there's a lot of really cool methodologies that you could do.
Yeah, and we did a lot of my most of my PhD honestly was spent on a machine called the flow cytometer.
Would you mind telling folks about the flow cytometer?
I will, yeah. So if we go back to antibodies for a second, scientists actually use antibodies all the time in our research. But what we do is we take an antibody and we link it to a fluorescent protein. So something, and if you guys have talked in microscopy before about fluorescent protein, so this is something that when a certain wavelength of light hits it, it will light up a certain color. So we know that on the surface of different immune cells, they have little flags. We call them markers. And what I can do is I can take if this is my CD4 cell, and this is my little flag that my CD4 is holding, I can take an antibody that has been labeled and I can have it so that it binds to that flag. And then any time this cell is made, I now can see it because of that antibody that's bound to it. So what the machine does is it's looking for the different colors of cells and it's sorting them according to the different colors. And it's detecting those different colors so that I can identify these cells and see how they're changing. So in a flow cytometer, the work that I'm doing is I'm trying to find my different kinds of immune cells, and then I'm measuring them. I'm looking to see, well, are they changing? Are they more? Are they less? Are they a different kind of cell now because they have different markers than they had before? And this technique allows me to do that.
It's a really, really helpful and elegant technique to be able to sort those cells. And then you could do a ton of follow-up on those individual populations, right? Without having to worry about well, what if I have a ton of different stuff in here?
Mm-hmm. Yeah, and that's what I would do for my bone marrow transplant experiments, is I would isolate out the exact cells that I wanted through this technique. They would get sorted into their own little tube, and then we would do the bone marrow transplant. We would inject them into the mice and then wait. Usually we waited like 4 to 6 weeks, depending on what we were doing, and then do follow-up experiments at that point.
Could you still see them?
Sometimes, yeah.
Cool.
It depended on what we were doing. Sometimes it didn't work because science is a failure sometimes. And then sometimes it did work, and it was really cool. Those were definitely the highlights on the days that it did work.
One thing you mentioned in passing was that you're working on mice and doing these bone marrow transplants instead of working on humans because of ethical concerns. Could you comment on just the importance of using mice as a model to inform us of humans? Like, why is it a great system to work on to get insights about ourselves?
Yeah, that's a good question. So model organism research in general is valuable because from an evolution perspective, we all have similar traits, we have similar properties, we have similar genetics. So mice and humans, even though we look very different from each other, we do have a surprising amount of genes that are the same. And because of that, we can study how a process works in a mouse, and that will help us figure out how this same process might work in a human. Now, of course, once you do an experiment in a mouse, you have to then do follow-up experiments before you eventually get to like something that can be given to a human. So it doesn't always translate 100% of the time. But the types of questions that we want to ask and answer, like if we want to develop a new medication or a new cure for a disease, you have to know how that thing works before you can then try to tackle it. My favorite analogy to this is like, if my lights in my house stop working, you don't want me trying to go fix the electrical panel because I don't know how the electrical panel is supposed to work normally. Like that would be bad news. But the same is true for anything, right? Like if we don't understand how the immune system works, then we can't try to fix it when somebody has an immune system disease. So by studying model organisms that have similar immune systems to us, like a mouse, we can learn how the immune system works to then try to fix what's going wrong in these human diseases.
So literally training on one and then moving over to it back to the human now and being able to correct it, if you will, because now you understand the fundamentals of it.
Right.
So really cool work on the mice immunity. What was your next step? So you've gotten your PhD. How did you decide what your next step was, and what was it?
So in my PhD, learned pretty quickly that I didn't want to run my own lab in the way that they were running their lab, the Jackson laboratory where I was. And it's not that I loved my time there, and it was a fantastic institution. It was more so that the scientists who were running the labs were in their office all the time writing grants. That was all they were doing. In fact, I mentioned how my PhD advisor, I was his last student, and anytime he had a chance to get into the lab, he was like a kid in a candy store. Like he would be like jumping up and down, like super excited because most of his time he was in his office writing papers and writing a grant. And I knew that I didn't want to do that. One is like, it just was stressful. It seemed I was like the stress of everybody was like exuding onto me because it's a hard environment, right? Because you have to publish, you have to write grants, you have to get funding, or you can't keep your job. So I figured out pretty quickly that I didn't want that job. I didn't want all of my job to be just in my office writing grants all day. And so I started trying to think about what other things could I do with my PhD, because I was in my PhD by this point. And that's where I started thinking about how I could kind of combine my love of education with my love of science. So as a grad student, I started teaching. I started making sure that I got teaching experience. I started doing outreach events in the community with like libraries and schools and those types of things. So when it came time for me to graduate after I finished my degree, I was trying to figure out what to do next. And to be totally honest, I didn't want to do a postdoc because normally you get a PhD and then you go do a postdoc. And that's like more years of specialized training. But everyone was like, no, you have to do a postdoc. And I tried to apply to jobs without a postdoc, and like none of them quite worked. Like I did get like an offer, but it wasn't didn't feel like a good fit. So I was just like, okay, I'm going to do a postdoc. But then I was like, if I'm going to do a postdoc, I've only ever worked with mice. This is a problem. Mice are great model organisms for immune system stuff, but they're not good for education because they're expensive and you can't, like, they're hard to work with. The undergraduate institution that I was at was it's very rare to have a small undergraduate state university that doesn't have a graduate program have mice in the first place. So I decided to use my postdoc as a way of learning a new model organism. So I interviewed for a postdoc with a scientist named Dustin Updike. He is a C. elegans scientist. So C. elegans are a tiny little roundworm. You can't really see them with the naked eye. You have to look at them under a microscope. And when I met with him, I was like, Dustin, I was like, I really want to do education. I was like, I am excited to learn a new model organism, but my goal is education. And he was on board with that. Not all PIs would be like that, but it was very important for me to be upfront with whoever was hiring me because I didn't want to deceive them, but I also didn't want them being mad at me if I was also doing teaching and stuff. So it was very important for me to be upfront. And he was 100% supportive. He let me go to 6 months into my postdoc, I essentially took 3 weeks off to go teach at a university for a winter term to get teaching experience. And he was very on board with that. So I worked with him for 4 years using this really great model organism. Like the C. elegans is amazing because it's very cheap. I can buy a whole plate of C. elegans for like $7, whereas the mice I was working with at Jack's were like $500 a mouse. So it's like massively different in price. And also it's clear, it's transparent. So I could do really cool microscopy and I could ask really cool questions and I could do live imaging. So I could do live microscope imaging and I could see cells dividing and I could see like when I was doing experiments, I could immediately see what would happen just in a live animal, which was really cool. So I did my work with him and it was fantastic. I learned a whole new model organism and I gained a whole new set of skills compared to what I had as a PhD student.
Do you have, or are you allowed to answer a secret preference for one of the two, a mouse or a C. elegans?
So
it's not so much. It's not so much a favorite organism, but whatever question you're asking.
Yeah, like if I was to go, like if I wanted to answer an immunology question, I would definitely pick a mouse. But if I wanted to do like cool microscopy work, I would definitely pick a worm.
I like that answer a lot.
Yeah, it's definitely, it's hard. It's like picking a favorite child though. They both are important and they both have helped, right? They both are really important model organisms for asking different kinds of questions.
What was it in your journey that led you to the conclusion that you really wanted to that you wanted to be an educator in the sciences? So like when you were, in your postdoc and you go up front, like, this is what I want to do, what had led you to that point that you really wanted to teach?
I think it was actually back down to like the impact that my teachers and professors had on me. And one of the things that I think about is like, I don't think I can ever thank those faculty members enough for we actually still talk. A couple of them came to my wedding. Like, they are amazing people. But I don't think I can ever like repay them for what they did. So one of the things that I always think about is like, how can I pay it forward? And I really think like, I have a Substack newsletter and I recently wrote a Substack about how science saved my life. And I definitely mean that literally in the sense that like, I wouldn't be where I am today if it wasn't for all of those people. I wouldn't have my husband or my kids because I met my husband while I was in grad school, all of those things. So I think for me, like as I was in grad school and I started like thinking more and more about what I value in life, I really realized how much education makes a difference in access. And that really, I think, was when I thought about where I could make the biggest difference, it ultimately came down to me that I think I can make a bigger difference through education than I could from leading my own lab.
So yeah. I guess that transitioned us really quite nicely to another thing that you're massive on is science communication, which is just education, right, for the broader community. Yeah. Could you tell us how did you get into being an Instagram influencer, a science hero and crusader on Instagram, and just like spreading the information information in a very similar way you did, that one-on-one kind of teaching?
Yeah, so I started this work in 2020, like March 2020, right around, what is it? Today's the 10th, right around now. 5 years ago, right around now. Wanna remember what that period of time was? It was the pandemic, right? So COVID had just been declared a pandemic. And at this point in time, I was a new mom. So my son was 8 months old, 9 months old. I guess no, actually, he would have been 11 months old when COVID was declared a pandemic. And prior to the pandemic, my husband and I had gone through a big journey with Owen where when I was pregnant with him, they told me that they thought he had a rare genetic disease. We were told he was going to have seizures, and they trained us on how to identify infantile spasms, which is this rare type of seizure that babies can get, but it kind of looks like normal baby movement because they flail, but normal babies also sometimes do that. So they had us watch videos, and we went through this whole process, and it was terrifying because I was a brand new mom. I was told when he was 4 weeks of age that he likely had this life-changing condition. And then I live in rural Maine. So the doctors were like, oh, we're going to send you to a specialist, but it's going to take 6 months to get there. And as a scientist, I was like, I can't do that. So because I was reading papers and I was doing all this stuff. Because when you're that's what you do that when you're a parent anyway, but I also have a science background. So I was reading every paper I could. And I got Owen into the, one of the best clinics in the country, happened to be at Boston Children's, which is not far from us. So I called and they got him in at 7 weeks of age. And to make a really long story short, the doctors in Maine were wrong. He didn't have this condition. He is fine. He's now turning 6 next month. And it took 6 months though for us to know. So the first 6 months of his life were very, very stressful for us. Because again, we were watching every move. We didn't have any other kids, so we were like, is this a normal baby movement or is this a seizure? What do we do? Luckily, the doctors at Boston Children's were amazing. They let us take videos and we could send it to them, and they would watch them for us to tell us whether or not we should be worried. They were amazing humans. Throughout that whole period, I was navigating this system, and it was hard. It was really hard to navigate this medical system. We had to get genetic testing, we had to get MRIs, we had all these things we were doing. And I had a science background. So I was able to read papers and ask questions. And I was able to, we got Owen, he almost got enrolled in a clinical trial because I found the clinical trial. We talked to them. We didn't end up doing it because they were wrong. He didn't have it. But my science knowledge, to make a long story short, really helped me navigate this situation. So I just started posting for my friends, explaining what was happening, doing all of that stuff. And then people started sharing it. And then I was like, I don't know if I really want people sharing my private Facebook page where the pictures of my almost 1-year-old are. So then I started ScienceWhizliz, which is the same handle I use here. I have here. And that's kind of just where it started. And for me, at the end of the day, I think it's about making sure people have access to information they can understand. Because if I hadn't had a science background, I couldn't have navigated any of the information that I was finding related to what they thought Owen had, because there was not very much good, there was information for parents, but it didn't answer the questions parents wanted to know, right? So it really made me appreciate the fact that access to science information, knowing where to find it, knowing what's true versus what's not true, none of those things are easy for people. So that really is what instigated it. And then since then, I've continued on with that.
Was this during your postdoc or after the postdoc that you started big time into the science communication?
I started my postdoc. I started while I was a postdoc, and then I actually signed my faculty position offer 2 weeks before the world shut down. So then I was like, am I going to have a job in September after all? I don't know. So I was transitioning from a postdoc to a faculty position when I started.
Do you feel like that I'm sure there's been a lot of complicating factors in being such a visible science communicator. I'm sure there's a lot of negativity that also comes your way, but do you feel like that you've been able to make a strong positive impact? Would you go back and do it again?
Yes. Yeah. Yeah. And I think for me, it comes down to the fact that for better or for worse, social media is here, right? We can argue all day about whether or not it should exist, but for right now it exists. And that is where people are going for information. And if scientists don't take the responsibility of communicating with the public, then honestly, I I partially blame us for where we are right now in history, because I feel like for a long time, and by we, I mean the collective body of people, not one individual, is right. We're in our ivory towers doing our research, being like, this is important, but we haven't necessarily taken the time or valued actually explaining to the public why it's important, or how we did it, or why we do it. All of those things that are important, because most institutions don't value public science communication as part of your tenure or your promotion. It's seen as like extracurricular activities that you do. And I think that has partially fed into the situation we're in now, where people don't trust science and don't value science to the same degree. And I think it's because we haven't done a great job of explaining why they should.
Yeah.
So I feel like I feel a very strong personal responsibility to try to change other scientists' mind on that, but also to do what I can to help with that.
That is a very similar perspective that I share. We had a question from the chat. I'm curious to hear your perspective on how science communicators can help combat science denial for the good of public health, given your unique position having experienced both sides of the issue.
I have a lot of thoughts on this. I actually, right before this, from 5 to 6 p.m., I did another talk with a scientist named Melanie Tracek King. She also combats misinformation, and we talked about this exact thing. So I have a lot of thoughts, and I feel very strongly that the way to do it is not through shame or bullying. And I say this because I see a lot of people who I think are understandably, life is really stressful right now. We're in a scary period of time in history, and people want to automatically blame and like shame people who think differently than they do. For example, the child who died of measles in Texas recently. All I saw have been like comments about those parents should be thrown into jail. I feel very strongly that that is not the way to change minds. And I've changed minds. And that is not how I've done it. And I don't think, and the research on this topic shows that that's not the way you do it, right? So I feel very strongly that it takes empathy and it takes building relationships with people in order to change minds, because it's not information alone that will change minds. And this is something I posted about recently on my Instagram, and I got some pushback from people who very much thought the parents should get thrown into jail for this. And I think they were like, well, the information's out there, so if they chose to do something different, they should be thrown in jail for that. But the reality is that assumes that everyone knows how to access information, and it assumes everybody knows what information is good and what information is not. And that is increasingly harder to do right now in this period of history, right? So there's a lot of privilege to say that, oh, yes, I can find the information. I know how to use the information. I know this information's bad and this information's good. And that's not a lot of people's reality, because at the end of the day, most parents want to keep their kids safe. And even my mom, she made the decision not to vaccinate me because this was in the 80s, and she went to her pediatrician. My mom didn't go to high school. She only finished 8th grade. She has low literacy levels. Like, she went to her pediatrician and she asked her pediatrician a question about a vaccine, and the pediatrician told her, you vaccinate your child because I told you to. And that's all he said. So she took me and she left, and I never went to a pediatrician again. I didn't go back to a doctor until I was like a teenager. And at the end of the day, she made the decision she did. I don't agree with it now, but she made that decision because she loved me and she wanted to keep me safe. And she felt like that meant not vaccinating. And I think if we come at these conversations from that shared, right, we might disagree on everything else in life, fundamentally. But most people, of course, there are exceptions, but most people really do want to keep their kids safe. And those are the way that I approach these conversations, is I come at it from a place of shared desire for their children and a lot of empathy, because it's hard to be a parent in the United States right now. It's hard to be navigating misinformation when it's everywhere you look online, now in our highest office, right? Like, it's hard. I don't know if I answered the question, but I think at the end of the day, it ultimately comes down to being willing to have hard conversations and being willing to be empathetic with people. And I have changed minds, but it often takes a lot of time.
And that's not just for vaccine or not. That's for any of these scientific principles, right? And just like you were saying, that ivory tower mentality of the scientists, the collective we. Why should I believe this? Because I told you to.
Right.
And the same as the pediatrician, the same about a data set. It can be easily seen as to why someone wouldn't want to participate in that.
Yeah.
And so I really like your take of just make everyone part of the narrative, bring everyone into the conversation versus just saying this is how it is. Do you have any suggestions on effectively communicating something like that? So if you are trying to convince someone or trying to demonstrate to someone that there is this alternative, is there a general approach that you like to take? Yes, the empathetic route of it, but is there you find if you show an image that's really helpful, or is it just case by case basis? How do you go about doing this kind of communication?
Yeah, I think it comes down to trying to understand what it is that somebody is worrying about. So for example, I've had conversations with people who, when it comes down to it, they've heard scary claims about, I don't know, pick any scary claim, right? Like that vaccines cause autism or something, right? So they've heard that scary claim and they want to keep their kids safe. So they were like, okay, maybe that means I don't vaccinate. But when I started having these conversations with them, a lot of the time it's just they don't know how to navigate everything, and they don't know how to figure out what information is good versus bad, or why the science we have says this isn't true and why they should trust that thing. So a lot of times it just comes down to having a lot of conversations and trying to understand what's the fear or the concern or the motivation behind what they're doing or saying. I think too, in general, one thing I and other science communicators have talked a lot about is we need to just normalize these kinds of conversations. So on social media, you're more likely to hear about somebody who had something bad happen to them than you are to hear about the hundred and millions of times somebody does something and it's fine, right? How many planes fly every day and we don't hear about them because they fly and they're fine. And we only hear about the times they crash. So it's trying to normalize these conversations about science and the good things that science does in our everyday life, so that people start to see yeah, by the way, I did vaccinate my kid last week and he's fine. You don't have to make it necessarily a huge, big conversation. You could just mention it in passing, right? Because usually we don't. So it's what ends up happening is that the more misinformation is repeated, or the more you hear a claim, the more likely you are to believe that claim is true, even if it's not. So right now on social media, you're hearing all these scary things that are not true. And people who are normally very logical will be like, I wonder if I missed something. Why is it all of a sudden everyone's saying this? Did I miss something? And then from there, it kind of can spiral down. So it really is, I think, about having these conversations and making these things just normal part of our existence, that we mention, hey, I did this, or hey, my kid was vaccinated, is fine, or hey, I took this medication. Isn't it cool that we now have it because of science, or whatever it is, that we make that more of a part of what we do.
If, let's say, you're that logical person who's sitting in front of a computer screen and you see this message in the social media and you wonder to yourself, I wonder if I missed this. What is your suggestion on finding and circumventing the misinformation to get to the correct, right? Because that seems like the really, really difficult thing right now. Is there a way for someone who maybe is, learning from a science communicator like yourself and now wants to follow up on something? Well, how would you help them? What would your advice be on how to navigate the current social media climate to get to the answer versus falling into what might be a conspiracy?
First and foremost, it's be skeptical. Everything you see, be skeptical. I tell my own followers this all the time. I don't want you to trust something that I say just because it's me saying it, right? You should go ahead and ask for sources. And I put sources on what I'm saying. If somebody doesn't put sources on it, you should ask them for it. And if they won't give you sources, that's a problem. They should be able to back up what they're saying if they're talking about something in science or medicine. So definitely be skeptical, and then go off social media to try to verify. So there's a technique called lateral reading, whereas let's say you find a claim somewhere, instead of trying to search on that platform or that website, open a new tab and Google that thing with fact check, or Google the website it's on with like bias, and see what information you can find that does or does not support whatever the claim is, right? Because it's, even if you see a good science claim, it's still important to like verify that that is actually true, because everybody is susceptible to misinformation no matter what side of the spectrum you're on. So there's those things that like we should all be doing. We should all be skeptical. We should all go off and try to verify things that we're seeing. And pause before you share. A lot of times when it's emotional, like you get angry, like that should be a warning sign to you that you should pause, because a lot of times truth is boring, right? And that's why misinformation spreads so much compared to truth, because misinformation is more interesting. It's more salacious, right? It's cooler. So it gets spread more. So if you see that, if you have that instinct to get angry because you're reading something, or it elicits a very strong emotional response, that should automatically pause. You should pause and then try to go verify that claim somewhere else.
And that doesn't matter the direction of the claim, right? It's just, as always, regardless, it's this having a healthy amount of skepticism.
Exactly. And for example, recently, like this has been in the news a lot recently because last week, President Trump made the comment about transgender mice. And I saw this claim about 4 weeks ago now, where a friend of mine was like, oh my gosh, how do they not know the difference between transgenic and transgender mice? And my first instinct was like, oh my God, this is another thing. Like, what is going on? And my first instinct was like to be mad and to share it. But then I was like, no, I need to figure out what is going on here. So I did some digging and I found that no, like they meant, like they were using the term incorrectly, but they did mean transgender because it was pointing to some very specific studies that were looking at the impacts of gender affirming care, but using mouse models. But they were giving male mice estrogen or giving female mice testosterone because they want to understand how these hormones work, both for normal people, like who identify with whatever they were assigned. Also for people who might be taking them for menopause treatment or might be taking them because, athletes incorrectly sometimes take these things, right? There's a lot of reasons why. But every single study mentioned how this work was important for transgender healthcare, which is 100% research that should happen. But everyone saying that it was transgenic mice were wrong. And these are scientists who I see now still saying that, oh no, he meant transgenic mice, right? And that's not what he meant. So anyone can have that instinct to spread misinformation. So it's really important that we check ourselves. And this matters because we should be arguing about why it's important to do this work for all the many reasons it's important, because transgender individuals deserve to have research that answers their unique medical needs, but also because this work will impact all of us in some way, because the more we understand about how these things work, the better. But instead, I still see scientists arguing he meant transgenic mice.
I think that's a really good topical example about the importance, like you said, of fact checking again, because it is like a great thing that you can end up doing, like a retweet on there and like just wanting to push it because it's sensationalized. But like you said, it's important to actually dig and see what the actual data is or what is being referred to. So Liz, as we round out the evening, because I want to be respectful of your time, of course.
Yes.
One question that we've been asking a lot of the guests that we've had on is, what does it mean to you to be a woman in STEM?
What does it mean to me to be a woman in STEM? I think it just means, that's a good question. I think it just means having the ability to ask cool questions and support other people who are in this area. And I think at the end of the day, that's what's important for me, is that I get to answer cool questions. I get to like help people understand science. I get to answer cool questions and hopefully make it easier for people to come after me.
Thank you very much, Liz. And thank you very much for doing the work that you do and advocating for science, and also just having those questions in place of being able to ask everyone those questions. I think that's a beautiful key message to be able to have. And we thank you for everything you do. And thank you for coming on the show and chatting with us and highlighting your journey in the sciences.
Thank you for having me.
Very much. We'll see you soon.